Okay, here's a quick summary of the lecture I gave on inositol phospholipid metabolism, focusing on the generation of the second messenger IP3. So here's a diagram from the Hancock textbook. Now, we've previously looked at these G protein coupled receptors, clearly bound to these heterotrimeric G proteins. Now, we previously looked at this complex here to activate adenyl cyclase through a GS subunit. Now, there's another form of this complex that can be involved in activating phospholipase C. Okay, and phospholipase C is an important enzyme activity because it gives rise to the IP3 molecule, which is an important second messenger, in the same way that cyclic AMP is an important second messenger. And we'll have and later on we'll look at the role of IP3 in calcium signaling when we do the calcium lecture. So previously we've been considering the membrane as a place where signal transduction takes place. Now, in this context, we're looking at the membrane also as a source of substrates for cell signaling. Now, there are various membrane lipids present in the cell, and some of these membrane lipids contain inositol sugar. Now, the inositol can be phosphorylated to give rise to a range of inositol phospholipids. And these phospholipids can serve as a substrate to be broken down to give rise to various second messengers within the cell. So common forms of these are phosphatidyl inositol, which we simply refer to as PI. So here's a phosphatidyl inositol here with a glycerol molecule, a diacyl glycerol because it's got two acyl groups attached to it, joined through a phosphate group to a inositol sugar. And the sugar has various OH groups that can be phosphorylated. So we have a range of different phosphorylated forms of these phosphatidyl inositols. So here we have a diagram from Hancock which shows that information. So here's the diacyl glycerol component of the phosphatidyl inositol. Here's the phosphate group that links the sugar to the diacyl glycerol, and here's a sugar shown phosphorylated on two positions. I think that's the 4 and the 5 positions. So phospholipase C is the enzyme we'll be looking at that cleaves the phosphatidyl inositol to give rise to a second messenger, typically IP3. And when you cleave at this part of the molecule, this region here is embedded in the membrane and stays in the membrane, and this region here is soluble and can go into the cytosol. Now, inositol is an important component of the membrane lipids, but you should be aware that only a small proportion of membrane lipids actually contain an inositol. And the majority of these inositol-containing membrane lipids are not phosphorylated. So effectively, the phosphorylated inositol-containing lipids are present in a very small amount. And it's these particular lipids that are the substrate for phospholipase C, and that can give rise to second messengers. Now here's a little diagram which shows the sequential phosphorylation of the inositol sugar. So in this case here we have a plain inositol lipid, and we have an enzyme activity PI4 kinase which phosphorylates on the 4 position, and we have a phosphodinositol phospho-5 kinase, which phosphorylates on the 5 position. So there's a series of sequential phosphorylation steps which give rise to these phosphorylated lipids. Cleavage of these phospholipids always gives rise to a phosphate on the first group. So PIP2 gives rise to IP3, and this phosphate group was previously contained within the molecule. So effectively, what happens when PIP2 is hydrolyzed by phospholipase? Effectively, it gives rise to this small soluble second messenger referred to as IP3. IP3 can diffuse from the plasma membrane, and it can interact with a IP3 receptor which is on the membrane of the endoplasmic reticulum. Now, the endoplasmic reticulum stores calcium, and IP3 allows the receptor to release calcium into the cytoplasm. So the effect of IP3 is to cause a sharp rise in calcium ion concentrations within the cytosol. Now, one of the effects of increased levels of calcium ions is the activation of protein kinase C. Now, bear in mind that when IP3 is produced, it leaves behind the diacylglycerol in the membrane. Diacylglycerol also cooperates with calcium ions to activate protein kinase C. Again, here's a good diagram from the Leninger textbook which shows the activation of a G protein coupled receptor by a hormone, leading to the activation of the heterotrimeric G protein. In this instance, we refer to the alpha subunit as GQ, and the GQ subunit, once it's bound to GTP, as shown here, can activate phospholipase C. Phospholipase C can cleave the phosphorylated inositol lipid, giving rise to diacylglycerol that remains in the membrane and releasing IP3 into the cytosol, where it can bind to these IP3 receptors, which are also calcium channels, allowing the release of calcium from internal stores in the endoplasmic reticulum out into the cytosol. And the sharp level increase of calcium ions leads to the activation of protein kinase C, and also protein kinase C activation is helped by the diacylglycerol. So the diacylglycerol and the calcium ions combine to activate protein kinase C, which can then phosphorylate other cellular components. So as we've already stated, the PIP2, which is the substrate for phospholipase C to give rise to IP3 and the diacylglycerol, the PIP2 is produced by sequential phosphorylation of these phosphatidyl inositols. Typically, there are various kinases involved in this process. Also, there's another enzyme activity which is phosphoinositide 3 kinase, which can phosphorylate and interconvert these various phospholipids into various forms. So there can be various forms of these phospholipids present in the cell. Now, bear in mind that PIP2 is a substrate for diacylglycerol and IP3. So phospholipase C cleaves PIP2 into these second messengers, but there's another enzyme activity that could convert PIP2 into PIP3. PIP3 is not a substrate for phospholipase C. So these various kinases can modulate the amounts of substrate for the phospholipase C, and therefore the various signaling pathways that can occur. So we've been discussing phospholipase C as an enzyme which is activated by the GQ subunit of the heterotrimeric G protein complex. I showed you that diagram a couple of slides ago. Now, there are various isoforms of phospholipase C, and there's another isoform of phospholipase C which has an SH2 group on it, an SH2 domain on it, and it can interact directly with receptor tyrosine kinases to become activated, also to give rise to the second messengers IP3 and diacylglycerol. So there are various isoforms of phospholipase C, some of which can be activated by G protein coupled receptors, and some of which can be activated by receptor tyrosine kinase. So we've discussed that PIC2 can be cleaved to give rise to IP3, but also other phosphatidyl inositols can be cleaved to give rise to additional second messengers. Now, these other soluble inositol compounds can be further metabolized in the cytosol, and this gives rise to a mishmash of different phosphorylated inositol compounds within the cell, which is currently being studied. So there's a lot of these molecules present in the cell, and their roles are becoming apparent. So here we have sort of a flow diagram showing the metabolism of these phosphatidyl inositols. So here's our PIP2, which we've discussed can be cleaved by the phospholipase C to give rise to IP3. Now, IP3 not only can activate the release of calcium through the IP3 receptor in the endoplasmic reticulum, it can also be phosphorylated or acted upon by a kinase to be additionally phosphorylated or some of the phosphate groups removed. And then these highly phosphorylated molecules can be further metabolized, giving rise to various compounds which may have an effect on some process in the cell which is either poorly defined or way too complicated to be described in the textbook. But it's just interesting to know that these processes occur. Okay, I hope that helps.